Okay, so today we're going to talk about part two of metabolism, which is the breakdown of carbohydrates. Um, there's two ways that this can occur, either through cellular respiration, which is something you guys should be familiar with, and that includes the pathways that are listed on this slide, which is glycolysis, the Krebs cycle, and the electron transport chain. Remember on the first part of this lecture, on the last recording, the electron transport chain is where oxidative phosphorylation occurs, which is one of the three ways to make ATP. The second way to break down carbohydrates is by fermentation, which is an anaerobic respiration pathway. And that will be in the third and final part of the metabolism recordings. So this little schematic here sort of takes you through your two options. You can either do respiration, which again includes glycolysis, the Krebs cycle, and the electron transport chain. You should know what each of those pathways is responsible for, what's occurring out of each of those pathways, what products are being formed out of each of those pathways, and how many ATPs and by which mechanisms those ATPs are being made, whether it's substrate level phosphorylation or oxidative phosphorylation. You have a brief description of these pathways, which are listed here in these little boxes. Something that you can, at least start off with getting an understanding of what's happening. Each of the products is sort of listed here as well, okay? Although these are not total numbers. You need to dive into the pathways a little more specifically to see the actual total numbers. There are also several review sheets that are posted on Canvas that go into this as well. On the right-hand side, we have fermentation. And as you can see, the pathways are much simpler, so that will make for a little bit of an easier time here. The big difference between respiration and fermentation is the availability of oxygen. The other difference is not all organisms have the ability to switch between respiration and fermentation. In order to be able to do respiration, you need to have the enzymes for all the pathways to do respiration. Same goes for fermentation. In order to be able to do fermentation, you need to have the enzymes present to be able to do fermentation. And not all bacteria have those. So looking at glycolysis, this is the oxidation of glucose molecule to pyruvic acid, in which you produce ATP and NADH molecules as well. Before I continue, I just want to talk about the electron carriers. We have NADH and FADH. Their job is to pick up electrons on the pathway and shuttle them to the electron transport chain where they will release them for oxidative phosphorylation. If an NADH delivers electrons to the electron transport chain, the electron transport chain, by oxidative phosphorylation, will make 3 ATPs. If an FADH delivers electrons to the electron transport chain, and the electron transport chain undergoes oxidative phosphorylation, it will make 2 ATPs. So in essence, an NADH is worth the equivalent of 3 ATPs by oxidative phosphorylation. FADH is worth 2 ATPs by oxidative phosphorylation. So when we look at glycolysis, the first thing that has to happen is we have to actually activate the sugar. And this is what we call the preparatory stage. And there are 2 ATPs that are used during this stage. So already you're actually expending 2 ATPs before you even make any ATPs. Those 2 ATPs will have to be subtracted from the total count of your ATPs. Glucose is then split to form 2 molecules of glyceraldehyde 3-phosphate. So if we look, this is glycolysis here. You do not need to know all the steps involved here. In the review sheets, specifically the the PDF of the one that I have handwritten out, the pathways, it's broken down into the stages that you do need to know, okay? So if we look here, during our preparatory phase, we can see that we're putting in an ATP and we're putting in a second ATP. And we're essentially putting phosphates on either end of the glucose molecule here. What then will happen is an enzyme will split this molecule in half. And it will form glyceraldehyde 3-phosphate and a molecule called dihydroxyacetone phosphate. Now the problem is that dihydroxyacetone phosphate, or DHAP, cannot continue down glycolysis. And what occurs is there's a fifth enzyme that will actually convert DHAP to glyceraldehyde 3-phosphate. So it's going to move this phosphate to this side, making this molecule. So you essentially will then have two glyceraldehyde 3-phosphates continuing down the pathway, the original one and the one that was converted from the DHAP. Then what happens is you will start to process glyceraldehyde 3-phosphate to make two pyruvic acid molecules, okay? In the meantime, you're going to end up producing two NADH molecules, two ATPs, and an additional two ATPs. So in total, you're going to make 4 ATPs, 2 plus 2, and then two NADH molecules. These two NADH molecules will then go off to the electron transport chain, where they will undergo oxidative phosphorylation, and because there's two NADHs, each one worth 3 ATPs, you will make 6 additional ATPs. So on glycolysis, if you add up the 6 by oxidative phosphorylation from the two NADHs and the 4 ATPs that you made by substrate level phosphorylation, 2 here and 2 here, you have a total of 10 ATPs for glycolysis. Now remember, we used 2 at the beginning, so your net gain would be 10 minus 2, which is a total of 8 ATPs, okay? You know, you want to spend particular attention to these notations that are here, okay? Because this is really pretty much going through what I just went through. And it's outlined here, the steps involved of glycolysis, okay? In addition, as I said, there are review sheets up there as well. Okay, so the overall, what you end up producing overall with glycolysis is listed here, okay, your products. You make 2 pyruvic acids, 4 ATP molecules, and 2 NADH molecules, okay? The overall net gain of 2 ATP molecules for each molecule of glucose is oxidized, plus the 2 NADHs, which will give you an additional 6. There are some alternative pathways to glycolysis. There's what we use called the pentose phosphate pathway and the Entner-Duderoff pathway. I'm not going to make you guys get into those, but these are alternative pathways to glycolysis. So with cellular respiration, what happens is the oxidation of molecules liberates electrons to operate on the electron transport chain. Now, the final electron acceptor comes from outside the cell and is inorganic, and ATP is generated by oxidative phosphorylation on the electron transport chain. So we've gone through glycolysis where we've produced 2 ATPs. I'm sorry, where we've produced 2 pyruvic acids and a net gain of 8 ATPs. That is under aerobic conditions. When we talk about fermentation in the third part of this lecture, we will go through glycolysis again. Glycolysis is a pathway that can occur both under aerobic and anaerobic conditions. The thing is moving forward from the pyruvic acid, going through the intermediate step, the TCA cycle, and the electron transport chain. Those are all pathways that must have oxygen, they only occur under aerobic conditions. So now that we've made pyruvic acids, 2 of them, by the Krebs cycle, by glycolysis, we will now go through the Krebs cycle. And what will happen is pyruvic acid is oxidized, meaning oxidation means broken down, and decarboxylation occurs. You're going to remove the carbons. The resulting 2-carbon compound attaches to coenzyme A, forming acetyl-CoA and NADH. Okay. The oxidation of acetyl-CoA then will produce NADHs, FADHs, ATP, and liberate the remaining CO2 as waste. So let's look at the Krebs cycle here. The first step is what we call the intermediate step. This is not technically part of the Krebs cycle. It is its own pathway in which pyruvic acid will be converted to acetyl-CoA. What's happening here is you are going through a decarboxylation step. You are removing a carbon, and that's coming off as a CO2. Whenever you break a bond, you release electrons. Whenever you release electrons, they have to be picked up by an electron carrier. And in that case, NAD picks up those electrons and we form an NADH. Okay. Now we have acetyl-CoA. Acetyl-CoA is going to get picked up by a molecule called oxoacetic acid. And essentially this molecule and acetyl-CoA will form a bond together and you will form citric acid. Citric acid will then quickly be converted to isocitric acid, which is its isomer. Once you have formed isocitric acid, you will then go through your first decarboxylation step of the TCA cycle. This will involve the removal of a carbon, as that's what decarboxylation means. So we break a bond, we release the CO2, which is the carbon. Because we broke a bond, we release electrons, they get picked up by NAD, forming NADH, okay? When we remove the carbon from isocitric acid, that will form a molecule called alpha-ketoglutarate, or alpha-glutaric acid. It's the same term. Alpha-ketoglutarate acid will then go through the second decarboxylation of of the TCA cycle. And again, we're going to break off a carbon. That carbon is going to come off as a CO2, okay? Because we broke a bond, we released electrons. Those electrons will get picked up by NAD, forming NADH, okay? Now what I want to point out something here is, remember that we have 2 pyruvic acids, so you're actually going to go through this process twice. And what I want to now point out is when we form succinyl-CoA by removing that carbon off of alpha-ketogluteric acid, what has now happened is we have released 1, 2, 3 CO2s. And if we're going through the pathway twice, that's a total of 6 CO2s, which means that the total carbons released is 6. If you remember, glucose is 6 carbons. So we have broken down glucose completely. The problem is we can't stop at succinyl-CoA because this is a cycle. And when we start with a cycle, we have to end where we started. So now the rest of the pathway is responsible for converting succinyl-CoA back up to our starting point, which is oxoacetic acid. But there's actually a lot of benefits to that. You're actually going to end up generating some more energy. And the first thing that's going to happen is we're going to get rid of our CoA. So succinyl-CoA will be converted to succinic acid, in which case we produce an ATP by substrate level phosphorylation. Succinic acid then gets converted to fumaric acid. You end up releasing some electrons in that process, which get picked up by FAD, forming FADH. Okay. Now, I always remember that the FADH comes into play because we're going from succinic acid to fumaric acid. Fumaric starts with an F, and that's where we form our FADHs. Fumaric acid gets converted to malic acid, and you get nothing for that. And then finally, Finally, malic acid gets converted back to oxoacetic acid, where you do release a few more electrons, which get picked up by NAD, forming NADH. Okay. Now, remember that you go around these, you go through these pathways twice. So all of your products are going to be double. Okay. The reason why you're going through these pathways twice is because you have two pyruvic acids. Now, what's going to happen to all the NADHs and the FADHs? They will shuttle those electrons that they picked up to the electron transport chain, where they will then undergo oxidative phosphorylation and produce ATPs. The NADHs will each give you 3 ATPs. The FADH will give you 2 ATPs. So that brings us to the electron transport chain. And the electron transport chain is a membrane-bound system. So the only membrane that we have in prokaryotic cells is the plasma membrane. And in a bacteria, that is where you would find the electron transport chain. In a eukaryotic cell, such as a human cell, the electron transport chain would be found in the inner membrane of the mitochondria. Okay, so again, it's a it's a membrane-bound structure. So the electron transport chain is a series of enzymes that are going to process these electrons, and you don't need to know all the cytochromes here. Okay. The process of making ATP from the electrons on the electron transport chain, as we mentioned, is called oxidative phosphorylation. Another name for that process is also called chemiosmosis. Okay, so make sure you know those terms. And what's going to happen is, the NADH or the FADH is going to release the electrons into this electron transport chain. Those electrons and protons are going to get transported through the chain. You're going to form a proton gradient on the other side of the membrane. And that proton gradient will then fuel ATP synthase and generate ATPs. Okay. So in a little bit more of a simpler terms, and I'm okay if you know the simpler terms, especially for the electron transport chain, our electron carriers, NADH or FADH, will release their electrons to the electron transport chain. You form a proton gradient. The proton gradient generates the energy required to function this big enzyme called ATP synthase. ATP synthase will then generate ATPs. Remember, for every NADH, you're going to get 3 ATPs. For every FADH, you're going to get 2 ATPs. Now the final electron acceptor, because remember, something has to accept those electrons. Under aerobic conditions, the final electron acceptor is going to be oxygen. Okay? And here is the total equation of what we just did for cellular respiration. We started off with a 6-carbon glucose molecule. We know this was cellular respiration because oxygen was used. And we needed 38 ADPs and 38 phosphates. Remember, you're making the ATPs from ADP molecules. This will end up producing 6 CO2s, which is your waste product of cellular respiration, 6 water molecules. These water molecules were made on the electron transport chain, along with the ATPs, and 38 ATPs. The 38 ATPs, if you go through and add up all the ATPs that you made through glycolysis, the intermediate step, and the citric acid cycle, if you add up all the ATPs you made by substrate level phosphorylation, and then take all the NADHs that you made and FADHs you made and add up the ATPs that they would make, that number would equal 38. Again, each NADH can be oxidized in the electron transport chain to produce 3 molecules of ATP. Each FADH can produce 2 molecules of ATP. Okay. and that is aerobic respiration, going through the process of glycolysis, TCA cycle, and the electron transport chain. Before I end part 2 of the lecture, I just want to make a little side note here. This is not an easy topic. This is a topic that you should have had in a prereq course and most likely forgot or blocked because it's something that is not easy. But you do need to know these pathways. You need to know why they're important. You need to know what they are producing, the ATPs that they're producing. You know, listen through this recording. Map out your own pathways. Use the review sheets that are posted. I would also recommend that, you know, in addition to listening to me, maybe look up on Khan Academy or some other reputable source, another description of cellular respiration. You know, the more that you're exposed to it, the more likely you are to understand what's actually happening. And as you should be doing, read the text. Your textbook is very good at explaining the steps involved here. And as I mentioned, there are several review sheets that are posted. The reason why I did multiple review sheets is if you look, the format is a little bit different. One is handwritten out. The other is more typed up into paragraph form. Students learn differently. So I try to give different views of how to absorb this material. Okay, have a good day, guys.